7.4. You're right. Yeah, and that's because they actually came not from common ancestry, because they had similar environmental protective pressures of actually being able to fly. But yeah, looking through everyone's answers, I think everyone got it. Yeah. All right, I'm gonna. Anyone has any questions? I'm gonna move on here. All right, so for topic 7.4. Oh wait, does someone have a question? All right, looks good. Yeah, so 7.4, population genetics. This is one that also has a few key terms. I'm also gonna screenshot this and put this in the chat. But this is just a selection from the CED. And yeah, you can just peruse this. I'll also keep in the chat. And also thank you so much for telling me to go slower. It's been a while. But yeah, if in case I'm going too fast or too slow, just let me know immediately. Because yeah, thank you. All right, so what is population genetics? So evolution is not really driven by selection, but it's also driven by random processes. And so while natural selection is one mechanism, there are still also some random ones. And some of them are mutations, genetic drift, which I'll go over, and gene flow. And these random events can have huge effects, especially in such small populations. And looking at this image here, it's essentially a simulation of the allele frequencies within a single population. So as you can see, it all starts with an equal distribution of all the colors of blue, yellow, green, pink, and red. But as the genes progress, due to whether it's just some random processes or just through a process known as genetic drift, the allele frequencies change over time, and they don't maintain their 20% ratio. And so, for genetic drift, is made up of two main parts, and that's mutations, that's genetic drift. So mutations are random changes in the DNA. I'm sure everyone has gone over some of that Unit 4 stuff, and it's just the random changes that occur. And some of the effects I want to talk about are the bottleneck effect, which is in biology describes a significant reduction in the population size, often due to a catastrophic event leading to a loss of genetic diversity. I'm sure we all know the famous example of the dinosaurs being hit by a giant meteor. That could be seen as the bottleneck effect. And for the founder effect is an example of a small group which colonizes a new area. And the reason I cite the Amish here is because the Amish are essentially a prime example of the founder effect in human populations. They went and established themselves in rural areas, and then they isolated themselves, leading to a significant reduction in genetic diversity. And now to explain this image right here, the bottleneck effect is basically when the narrow passage would represent a sudden event like a natural disaster, filters out only some individuals into the new population, and has fewer dots and less color variety. But on the right side is just the founder effect, where the few individuals from the larger population migrate, which is what the arrow is showing, and then only contain a small subset of the original genetic diversity. And so it has fewer colors and lack alleles present in the original generation. All right, so next up is gene flow and variation. So gene flow is it's just another word for migration, is the movement of alleles from one population to another. And this increases genetic variation and makes populations more genetically, bottlenecks and the founder effect all reduce genetic diversity. And so, let me know if that makes sense. And basically back to gene flow though, as they migrate, it changes the allele frequencies of both populations. As seen in this butterfly example, population A and B all have a change in their allele frequencies. And it's important. It's like a genetic gene flow is an important counterbalance to genetic drift. And okay, well, would the founder effect be considered a part of gene flow? Honestly, I feel like that's just a matter of semantics, but I think it would. Is it okay to show which topic this covers as well? I will be covering topic 7.5, Hardy-Weinberg, later. But yeah, I will post this entire thing later. But yeah, in terms of whether the founder effect would be considered a part of gene flow, I think it would be, and if the founder effect established a new population in an already new population. But if they just went and established themselves on oh, I see what you mean. Don't crop the whole thing. Oh yeah. like by don't crop the whole thing, do you want me to send like this entire thing? Here, I'll show you. Like this right here. Oh yeah, okay, all right, I'll keep, yeah, I'll do that next time, yeah. But yeah, sorry to keep jumping between questions. But yeah, I think it would be considered gene flow if they went and established themselves in a newer population. But if they're just going to some random island that they're already alone in, then I think that would not be considered gene flow. Does that make sense, Greens? All right, perfect. All right. I realize now that the timer, I'm not going to set a timer this time. I'm just going to read the MCQ and hopefully that you guys answer in the chat. So a small population of birds is blown away to an island over a storm. Oh yeah, all good. Yeah, you can have a good night. Over generations, certain alleles become more common due to chance, and this is an example of A, natural selection, B, mutation, C, genetic drift, and D, gene flow. Yeah, I think everyone's got that. Yeah, so C. And yeah, this is just a definition example, but the correct answer is genetic drift since the change wasn't due to selection but random chance. Anyone have any questions about that? If not, oh, gene flow is when organisms migrate. It's just another synonym for migration. As you can see, gene flow is when the white butterflies here move to the pink butterflies. So I do see its confusion that it could be gene flow because they're technically migrating, right? But the key example is that they're colonizing a new area. And perhaps this can't be a good question, but gene flow is more when an organism from a certain population moves to another population. While oh my gosh, wait. Yeah. Oh wait, oh my gosh, wait, I'm super sorry, guys. Yeah. This actually would be an example of gene flow. I don't know why. But yeah, you guys are totally right. And the reason for that is, yeah, well, it's not genetic drift because, oh my gosh, I'm reading off the wrong presenter slides right now. Wait, what is the answer for this one? Oh, so yeah, the key here is that over generations, certain alleles become more common due to chance. And I think that the initial part, the small population of birds being blown away to an island, that would be gene flow, but over generations, certain alleles become more common due to chance. That would be genetic drift. So I do see where you're coming from. So I think that this question I honestly just got from Quizlet wasn't too good. But I would actually say that this first part where a small population of birds is blown away, would be gene flow. Over generations, the certain alleles becoming more common, that would be genetic drift. Yes, yeah. Going to CS7 AP's test this year. That's a great summary. Yeah. Sorry about that, guys. Wouldn't it only be gene flow if there is a population of birds already established on the island? Yeah, it would be. Like I said before, it's kind of implied. Like I said, I kind of actually want to revise this question. Maybe I want to take out D actually from this question. But that would be true. It's only gene flow if there's migration happening between populations. But yeah, sorry about that, guys. I was reading from the wrong. I have another slide here telling the answers, and I got skipped up on the next slide. But yeah, anyone have any questions about that? All right, if not, I'm going to move on to the next slide. Yeah, well, hopefully I'm not learning right now. But all right, so 7.5, I'm also I'll do what you said, and I'll post the entire CED here. But as we're going through the slides, you can look through this. We're going to Hardy-Weinberg now, so it's going to be a lot of math-based. And although I recommend you have a calculator, all the math problems I did in this section will not require a calculator and can be done in your head. Alright, so in case you read this, I'm going to move on to the next slide. So, alright, so what is the Hardy-Weinberg model? So the Hardy-Weinberg model assumes 5 conditions. And these conditions are often not present in actual populations, but they're just here for prediction stance. So these 5 conditions are no mutations, random mating. Random mating means that basically there's no preference for one genotype over the other. There's no natural selection, a very large population, and that means no genetic drift, and there's no gene flow. And oh yes, okay, so you will actually have to memorize these conditions, sadly. I did remember there was an FRQ that I actually think I did. I took the exam last year, and I believe there was an FRQ. There was an FRQ talking about that required you to know these like on the dot. How I remember them is mutant rats never love grapes, and you can make your own mnemonics, but I remember this as no mutations, random mating as mutant rats, and then no natural selection as never, large population as love, and no gene flow as grapes. So, I mean, you can make your own mnemonic, but if that helps you, then I'm glad. I'm glad that mutant rats never love grapes. Do questions ever trick you? Like, oh, recessive is 36%, and by the way, there is natural selection. I've never seen a question like that personally, but I guess if you did see a question like that, then you would automatically know that Hardy-Weinberg does not apply, and you can't use any of the equations. The second point in essential knowledge means, if you're talking about allele frequencies in a population can be calculated by genotypic frequencies. Are you talking about this one here? Here, I'll put this here. Are you talking about that point there? Okay, you don't have to memorize the equations. I believe there are, well, they're not that too hard to memorize, but I believe that they're also on the equation sheet. Anch, is that the point that you were talking about? The small population. Okay, I'll get to that in a few more slides. But essentially small population sheets are more impacted by random events. So actually I'll talk about that now. So large populations means less genetic drift because larger populations are less impacted by random events. Let's say in a population of 5 people, let's make it a lot smaller. In a population of 3 people, each person having a certain allele. Like, let's say one person has allele A, one person has allele B, and one person has allele C. If one person dies, then that's 33% of the genetic variation gone. Like, originally it's 33% A, 33% B, and 33% C. But if the person with allele B dies, then now it's just 50% A and 50% B. But now, if in a large population, it was 300 people, 100 people had allele A, and 100 people had allele B, and 100 people had allele C. If one person from allele B died, that basically would not change anything. And that's the random event that is being talking about. That's the random death and loss of an allele. Does that make sense? All right, perfect. But let's see, did I miss any equations? Yes, you got it, exactly. Yeah, mutant rats never love grapes, guys. But I will be talking about this more and how to actually solve these equations. Net is equal to random. Net is like when I said net loss, it's just in general the loss that, like net loss means is like, in total, if we lose one allele from B, only a couple of the, instead of it being 33% A, 33% B, 33% C, it's now 31.9 or something. What's an example problem where the assumptions are required for us to know then? I can bring an FRQ, but basically they'll trick you with saying an MCQ that has a population, and it's going to be like true or false. Well, not true or false, but like, does this population apply to Hardy-Weinberg equilibrium? And in that case, that's an assumptions where these 5 assumptions are required for us to know. And how would a large population have no NS? Oh, how would a large population? Okay, yeah, and that's the thing. These conditions are likely never able to be possible. Hardy-Weinberg model is never used in. Well, I want to say never used in real life, but it's true. The natural selection always happens. Populations are always evolving. So a large population would always have natural selection. Yeah. Yeah, it's like nature's utopia. It's the perfect example. All right, so also please explain this reduction generation can increase the difference between populations of the same species. Is that from the CED or is that from the my slides? Because if it's from the CED, then I'll cover that in a sec. But yeah, it's like nature's utopia. Yes, it is. Yeah, and models aren't perfect. Yeah, that's completely right. Yeah. For Henny, is that from the CED or is that from my slides? Since if it's from the CED, I'll cover that in a sec. All right, I'll probably just well, if just bring that up again. Okay, I'll wait for you. All right, but so I'm sure. Okay, I got you. Yeah. You can stick around and hopefully we can after the lecture and hopefully we can I'll try to decipher what your teacher meant. So these are the equations. Yeah, the cool part. These equations are honestly pretty. Okay, so this is funny. This is really funny. If you have taken pre-calculus, you know that P plus Q is equal to 1. These equations are identical. P plus Q squared is P squared plus 2PQ plus Q squared. If you didn't understand what I just said, then you can ignore that. But that was a fun fact for anyone who's taken math. And yeah, who knows, guys? If we square it again, you can name the equation after yourselves. Like, who knows? Yeah. Yeah, okay, yeah. I think it might be algebra 1. But yeah, I just thought it was funny that this guy named Hardy Weinberg just squared an equation and just said it was his. But alright, so the two equations are P plus Q is equal to 1, which represents the allele frequencies. And by allele frequencies, I'll cover this later, but I'll cover it. Well, I'm going to go through practice problem later, but P represents the number of dominant alleles. And keep in mind, it's the number of dominant alleles. And it's not the person that expresses the phenotype, right? It's the number of recessive alleles. Sorry, number of dominant alleles. And Q represents the number of recessive alleles. And I want to emphasize, because it's such a common misconception, it's the alleles. It's not the amount of people that actually express the phenotype. And P plus Q is equal to 1. P squared plus 2PQ plus Q squared is equal to 1 as well. So P squared, and keep in mind, this is probably the biggest misconception with this. P squared is the number of homozygous dominant individuals, and Q squared is the number of the homozygous recessive individuals. And 2PQ is the number of heterozygous individuals. This is something that I feel like a lot of people are going to have a lot of questions on. So if you have any questions, just let me know, because I feel like that's just this is pretty confusing. All right, so I'll answer this question now. So wait, could I do P plus Q? Okay, yeah, you could, but you always want to use the expanded form since that's the only form that would actually work. But yeah, I wouldn't do that. Yeah. So if it's squared, it's the amount of people, and when it's normal, it's alleles. Yes, and that's exactly it. It's not the other way around. Okay, so if no more questions, then I'll move on to the next slide. So, how to solve Hardy-Weinberg problems? So this is where the key we wanted to get to, right? So, in order to solve these problems, you have to identify the known genotype frequencies from what they're giving you. So, it's basically just a word problem disguised as a biology question. You have to figure out and write down everything you know. You have to start by figuring out whether you're being given allele frequencies or genotype frequencies. And what that mean is, are they telling you there's 5 people who are red, or are they telling you there's 5 people who have the recessive red allele? And there's common keywords for this, and that's like recessive, dominant, homozygous. Those are all keywords you should look out for. And the reason that you should, okay, so the reason that you always want to solve for Q before P is because Q is often the recessive one. And the recessive one is always, basically heterozygous organisms always display the dominant trait, right? And because let's say that someone has a dominant allele and a recessive allele, they always display that dominant trait. And if they say let's say that red is dominant and blue is recessive. If they say 50 people have the red trait, and that's the dominant trait, then you can't know if they're homozygous or heterozygous. But if they tell you that that there's 10 blue people and blue is recessive, then you know for a fact that those people are homozygous recessive. Does that make sense? All right, perfect. And then so almost always, I said oftentimes, but there's never a single problem where you're not using it. You have to use the relationship P plus Q is equal to 1. And if you know what pure Q is, then you know what everything else is from the equations. And also, I also want to discuss this briefly, but sometimes there's a third allele, and we'll actually go over that at the end, actually, because I've never seen a problem like that, but someone in my previous lecture said they saw a problem like that, so I just added it onto the end of this slide, but I'll cover that later. All right, so time to test. Hopefully, if you have your calculators, you can solve this, but again, I won't start the timer, but all right, so yeah, in a population, 36% of the individuals show the recessive phenotype, and what is the frequency of the heterozygous genotype? All right, so it's, as a hint for this one, if the keyword is here is recessive G phenotype, and again, back to the question, why do you always solve for Q before P? And yeah, answer choice is A is 0.36, B is 0.48, 0.32, 0.24. And this is a little difficult. I will be giving a full explanation as to why this is after. Oh, congratulations, leveling up. But yeah, sorry, if this is confusing, I will be covering this exact problem and how to solve it. Yeah. In case you can't do the full math, you can just grab the equation. I believe that in order to give spoilers, it's double. Here, test. Yeah, it's double brackets. It's like two absolute values, or for my calculus guys out there, it's the magnitude sign. Oh wow, tons of people are leveling up from 4 to 5 here. Glad to hear that, guys. At EXP Farming. All right. Looks like everyone is saying B. In case that was confusing, I'll just give the. All right. So the solution to MCQ, right? So first of all, you know Q squared is equal to 0.36, and you know that because they told us 36% of the individuals show the recessive phenotype, and that's why Q squared is 0.36. By taking the square root of 0.36, we get 0.6, and using the equation P plus Q is equal to 1, we can calculate that P is 0.4. And then in order to get the frequency of the heterozygous genotype, we do 2 times P times Q, which is 2 times 0.4 times 0.6, which is 0.48. Does anyone have any questions with that? I know it can be a little hard. All right, I shall wait. Okay, can we solve one more question, actually? All right, hang on, let's see, did I? All right, we did not solve another one, but I'll type one in the chat right now. All right, so I'm just gonna, basically, all Hardy-Weinberg questions are usually always just reworded examples of the exact same problem. So I'll just type one out right now. Generate a Hardy-Weinberg problem. All right, let me see. Sorry, I'm just making sure that this problem is able to be done in your head, because I don't want you guys to pull out your calculators for this. Okay. All right, so let's say that B is dominant and lowercase b is recessive. Hashtag Thumbboard. Wait, is that the whiteboard that you're talking about? Is that what you're talking about? Oh, yeah, I've seen those, yeah. Okay, alright, so I just typed this up. Hopefully, I'm pretty sure you can do this in your head. If not, then I failed at life. But I typed this in chat. In a population of beetles, body color is determined by a single gene with 2 alleles. B is black and lowercase b is recessive. Researchers found that 50% of the population was brown. What is the frequencies of the capital B and lowercase b alleles? Alright, I didn't give MCQs since I didn't have time, but I'll give it around 1 minute, see if you can solve that. Oh shoot, okay, I'm solving this right now. You actually can't do it in your head. But just leave it as an equation form, and I'll just tell you if it's right or not. Or you can round it, yeah. So, to give a hint, you always want to solve for Q, and then just solve the equations from there. How will a question be worded if you start with Q? What about Q squared? Okay, so if it started with Q squared, then they'll tell you how many recessive individuals there are, and if for example, they'll tell you that there's 20% or 20 out of 100 individuals have the express the recessive phenotype, and if it's just Q, then just tell you how many Q alleles there are. Does that make sense? And it's kind of like in the original problem here. The keyword, if they start with Q squared, is recessive phenotype. And so for how a question will be worded if you start with Q squared, it's mostly they'll say recessive phenotype, or it's implied that it's the recessive phenotype. I'll start unveiling the spoilers right now. All right, perfect. Okay, so yeah, everyone who says, I think everyone has got it. I'll actually give some more time, actually. I'll give some more time. All right, I think that, yeah. Okay, so a mistake that you don't want to mistake is accidentally using the wrong equation. But yeah, a common mistake would be to use the 2pq equation when actually you're just solving for p and q. Yeah, and so you just want to figure out what p and q are, so it's a little different in that way. And the answer is approximately B is capital B is approximately 0.293, I think, and the recessive lowercase b is approximately 0.7. So yeah, let me know if you guys have any questions. Alright, alright, perfect. Wait, Henny, does that, Henny's emo girl, does that make sense? Or, alright, well, with the AP exam, alright, perfect, yeah. With the AP exam actually just asks us to calculate. Depends on if it's an FRQ or an MCQ. If it's an MCQ, they're gonna ask you which one is the closest to the genotypes, and they'll give you a list, like in my screen share right here. They'll tell you what's the closest answer. But for an FRQ, I actually see what you mean. Would the AP exam actually just ask us to calculate? um, yeah. So when I took the FRQ and I looked at the rubrics, you can just write the answer. I believe. This is going deep into my memory stores right now. I mean, I wouldn't see why you wouldn't just show your work, but I believe you can just write the correct answer. And how common is Hardy-Weinberg on FRQs? Okay, so here's the thing. You can look at a prediction. I believe that it's pretty common. It's almost it will be guaranteed to be on your test. I'm not sure about the FRQs, since it is literally like it's like the only math portion of AP biology considered other than the logistic equations, and the ICRT equations, I think. So I think it's pretty common on the FRQs. What's the predictions for this year's FRQ? Oh, shoot, I mean, you'll definitely find someone on YouTube definitely just scheming it out, but you can just look it up. I think AP Bio predictions. But yeah. All right, so if you don't have any questions, I'm gonna skedaddle on. All right, so time for topic 7.6, guys. We're halfway there. I'll post the CD again in the chat. You guys can read it while I'm screenshotting it. And I'll go over everything in the slides, but if you have I just post the CED here just as general overview to make sure that this is from the actual AP course, and so you're not learning extra. But all right, I'll move on. All right, so for the first part of the CED, you're just gonna want to know that this is on the CED, but personally, I've taken 4 practice exams, and I've taken and I've helped people with so many practice problems. The geometric dating, I've never seen an MCQ that's tested it, but I don't see why it wouldn't hurt to know. But essentially, fossils can give us a snapshot of previous life forms, including transitional species, and bridge the evolutionary gap between one species to another species. And we can date these fossils using some relative dating, and you won't need to know this about how exactly radiometric dating works, but it's done using half-life. But basically, by knowing the age of the rock where it's found in, you're able to figure out how old the fossil is, and thus how old the species was when it went extinct. And also tells you the geographic distribution of that species. Alright, and then, so DNA sequence similarity can also show relatedness. So, DNA protein sequences comparisons reveal how closely related species are, and the more similar the sequences are of their DNA is, the more recent their common ancestor is, and that's how more related they are. And so molecular evidence often provides more accurate than morphological / visual evidence, and that's mainly because of, like I said before, convergent evolution. Similar looking traits can, like bats and birds all have wings, but that doesn't mean that they're 100% related. But yeah, that's the 2 key things. And so these are back to the gold stuff. The homologous structures, homologous, refers to similar structures within organisms, but they share different functions, while vestigial structures are just remnants of features from ancestors, and examples of those in us are our tailbones, our appendix. And I actually had no idea about this. Yeah, okay, so here's the thing. Vestigial structures are 99% of the chance useless, but they can still be used. Like, if we lost it, we wouldn't die, but they're still, functioning, if that makes sense. Like, take the example of goosebumps. We wouldn't die if we didn't have them, but if they were gone, we wouldn't die, but they're still there, if that makes sense. But yeah, lastly, this relates to analogous structures, if you remember that from previous slides, because basically there's the 3 different types of structures are homologous structures and vestigial structures here. But analogous structures caused by convergent evolution also are related to this. Alright, so this is another pretty basic MCQ. So, which of the following best supports the conclusion that 2 species share a recent common ancestor? They live in similar environments and exhibit similar behaviors. Their embryos share similar development pathways early in development. And they have analogous structures which are suited for similar functions, and their physical appearances are nearly identical. Alright, yeah, everyone's got it, and I see people are leveling up too. Yeah, so the reason it's B is because especially shared development pathways, are considered strong evidences of common ancestry, while appearance and behavior can be misleading due to convergent evolution. That's why, and the reason C is wrong is because convergent evolution, and the reason why C and D are wrong is because just looking at stuff can't really tell you the relatedness, because it's not really quantitative, and they can just have evolved those structures in different areas, not as a result of common ancestry. All right. Any questions about that? But yeah, the answer is B. All right. I'll wait a little bit more. I see some people are typing. Doesn't analogic structures don't point to a common ancestor? Yes. Yeah, and that's exactly right. Yeah. So, analogous structures are caused by convergent evolution. And when we say that some two organisms have analogous structures, that doesn't mean that they're related. They just kind of look similar because of their analogous structures. Yeah, similar selective pressures. So it doesn't point to common ancestry. It just points to that they face similar selective pressures. And just to recap, selective pressures are similar threats that were posed to them. Does that make sense? Oh yeah, of course, yeah. 7.7 common ancestry. All right, so this is another topic where you just have to memorize 3 things. I mean, I'll post this in the chat. Introns. Okay, so introns. I'll go over that briefly, but that will also be covered in a separate unit review, because I think introns were a previous unit, and this is all about unit 7. But for now, yeah, I'll cover it in my slides. But so all eukaryotic organisms have membrane-bound organelles like the nucleus and mitochondria, and these organelles compartmentalize specialized functions. And like, for example, transcription in the nucleus, which I think that's a farther away unit, and ATP production in the mitochondria, which is the powerhouse of the cell, mitochondria. But essentially, the key point to take away here is the fact that all eukaryotes have these organelles, and that prokaryotes don't, which suggests that these features were inherited from some sort of shared common ancestor. And that's the fact that these have membrane-bound organelles. That all eukaryotes have membrane-bound organelles, which is why that they have a shared common ancestor. All right, I see someone's typing. Any questions about that? Not, I'll move on to the next slide. So what's an example of a problem where the assumptions are required for us to know them? Okay, so I took preparing this lecture, I also took another practice test, but they're gonna ask you oh no, it's totally fine. I'll ask that again. I'll answer it again. A direct MCQ question that could ask that is they'll just directly test you. Like they'll literally just say they'll give you 4 answer choices on MCQ, and they're gonna ask which of the following is evidence for eukaryote common ancestry. And they're going to be like, phospholipid bilayer, which is wrong. They're going to be like something else. And then for the answer, they'll be like, they'll just directly test you. For this one, there's not really much application you can do for this one. It's more like directly test. Does that make sense? All right, perfect. Could you explain why eukaryotic unicellulars can exist but prokaryotic multicellulars? Can you explain why eukaryotic? Wait, I feel like that. Are you asking why eukaryotes are? Well, I feel like that you got kind of mixed up. Eukaryotes are usually multicellular organisms, while prokaryotes are usually multicellular organisms. Like, is that what you're asking? Explain why eukaryotes are unicellulars but prokaryotes are multicellulars. Oh, okay, I see. you're asking why eukaryotic unicellulars, they do exist, but prokaryotic multicellulars can't. I mean, I'm honestly, in a whole, I feel like that it has to do with the main function that eukaryotes have a nucleus, but prokaryotes don't. I honestly don't have an exact answer for that. Maybe the person doing the Unit 4 review has an answer, but I don't. Sorry about that. Yeah. All right. So for membrane bound, so another piece of evidence that suggests is all eukaryotes have linear chromosomes, but prokaryotes have circular chromosomes, and that's a difference that also should suggest that all eukaryotes have a shared common ancestor. But and genes with introns. Okay, so I know that someone was asking about what's introns. I think that's a Unit 6 thing, I believe. And I will be going over Unit 6 next week. But introns are essentially the non-protein coding regions of genes. And what that means is that all DNA codes for amino acids, but some don't. If that makes sense. So for example, some code for amino acids. Some DNA sequence code for amino acids, but other DNA sequences code for transcription factors, which are essentially transcription factors or like some other stuff unrelated to proteins, and that's what our introns are. But yeah, I see what you mean. Yeah, that's why Unit 6 usually comes before Unit 7, but I did Unit 7 first. But so sorry about that. But next week I'll cover Unit 6, and hopefully this will make more sense then. But yeah, basically, when you do learn what introns are, they're another reason why eukaryotes have a shared common ancestor. All right. So, any questions about that? Okay. So yeah, I'll briefly I'll briefly explain it. So, introns are essentially again, this is a full subtopic in Unit 6, so I won't go too deep into it. But essentially, introns are the non-protein coding regions of genes. Sorry, non-protein coding regions. Yeah, non-protein coding DNA segments in genes and mRNA. And basically that means that they're not needed when transcribing a DNA. And yeah, sorry. I'll cover this more deeply in depth next week because introns are a big part. Yeah, but the non-protein coding regions are removed from the gene while like Greens is saying, exons are the coding segments which are kept inside the gene when transcribed. But yeah, next week I'll be covering Unit 6, which is probably one of the harder units, and this will make more sense then. Yeah, of course. All right, so I actually kind of want to skip this MCQ because I thought there were a couple people here that don't know too much about introns, so I'll actually skip this one. But I'll still leave it up for you guys to peruse. All right, I'll move on. If you have any questions, you can look on the slideshow. I'll post the link later. All right, so continuing evolution, I'll post the CED and the chat. All right, so the subtopic is just continuing evolution, and that's that all populations continue to evolve, and there's rarely a population when it's not evolving, and this ties back to the Hardy-Weinberg equilibrium. A population never has Hardy-Weinberg equilibrium, right? Our populations are always evolving, and like they're always evolving. So it is contradicting it because Hardy-Weinberg is someone had a good example is in Nature's Utopia. It's perfect, but populations are always evolving, and so populations are have evolved and continue to evolve, and some ways that they do this is through genomic changes over time, and that basically just involves genetic mutations, gene flow and genetic drift, which I've talked about before, and continuous. The reason I don't have a major slide for this is because I've kind of already covered this, but continuous changes in the fossil record, as I said before, they can be dated, and evolutions of bacteria's more resistance to antibiotics, pesticides, herbicides, and chemotherapy drugs show that bacteria are evolving over time. And the fact that pathogens are evolving and are bypassing our vaccines also shows that they're evolving. Are there any questions about this slide? I thought this was the most simple subunit, so I didn't go too depth into it. Alright. Yeah, alright, perfect. Next up, okay, alright. This is probably the hardest subunit, probably harder than Hardy-Weinberg. So I'm actually gonna before I go deep into depth, I'll allow around 30 seconds for you guys to just read over the CD to make sure that you guys know exactly what you need to know and what you don't need to know. And I'll also post this in the chat. But I hope. Yeah. Is that yeah it is, yeah. But this specific subunit I personally thought was one of the harder ones. Alright, well, morphological definition is relating to the form or structure of things. In biology, it's basically just saying that it's the exact definition of morphological is the form and structure of things. In terms of biology, it's usually used to refer to morphological structures. And that's saying like birds and bats, they have morphological structures, similar morphological structures, because they have wings. It's basically another synonym for like looking. Like they have similar looking structures, they have similar morphological structures. Does that make sense? Okay, yeah, I'll actually cover that. Yeah, I just allowed them. I'll cover all this. I'll cover the difference between cladograms and phylogenetic trees in their own slide. Homologous structures are essentially similar looking structures in two different organisms. Yeah. Alright, so hopefully that, well, the CD's in the chat in case you guys want to read it again, but here, I'll move on. So, phylogenetic trees and cladograms both show evolutionary relationships between two different species. And then the shared derived traits help determine the relationships. Hang on, what's going on here? Alright, so the shared evolutionary relationships, derived traits help determine relationships, and the nodes represent common ancestor. And I'll talk about shared derived traits in a sec, but before we talk about that, phylogenetic trees are essentially trees which show the branching between organisms. Does it have to be different function for homologous structure? It does not have to be different function, but it's literally 99% of homologous structures share a different function. The key is that homologous structures aren't caused by similar ancestry. So they just kind of look the same, but they usually have different functions, but they sometimes have the same function. But the key thing there is that the key takeaway for homologous structures is that they're not evidence of common ancestry. But anyway, so wait, sorry, does that make sense? Wait, alright, hang on, let me make sure I'm not messing up here. I don't want to, you're right, I don't want to be selling. Similar structures, different functions. And the vestigial, okay, so analogous structures, yeah. Oh my gosh, wait, am I? Shoot. Let me look at my screen for a second. So analogous structures are caused by convergent evolutions, but oh my gosh, yeah, you're right. Yeah, I'm super sorry, guys. Yeah, homologous structures are caused by our similar physical features. Yeah, okay, I'm sorry. Yeah, so they have different organisms, but they share a common ancestor. But analogous structures, that's the convergent evolution one. All right, yeah, sorry about that. Super sorry. So back to the slideshow. The nodes represent the common ancestors, but the outgroup is the most distantly related groups for in general comparison. So as you can see in this diagram here, the node is where the green and blue branches, but the C is the outgroup. And the outgroup is just the most distantly related organism. And so for what is phylogeny? It's just the physical characteristics. And we can build trees using morphological traits like bone structure, but molecular data such as DNA or protein sequences is more accurate. And that's because similar traits can evolve independently in unrelated species, and that's basically convergent evolution, which can mislead us if we only use physical features. And the false calibration can also add to the timescale of these trees. Alright. So another key point to take away is that phylogenetic trees are hypotheses, right? They're not an exact science, and that's a key point in the CD that they do want you to know. And then they also show speciation within a population over time. And then these trees can change with new evidence because they're hypotheses. Alright, so here's where it gets a little hard. Cladograms, they show relationships, not exactly time. Phylogenetic trees, they show the exact times that things branched, well, the exact estimates, but they can't be exact. But as you can see on the right, there's a phylogram, which shows the relative time for when things branches. While cladograms, they don't show the general time. They're more like just general relationships. Okay, and so this isn't on the CED, but I've seen this on my own FRQ when I actually took the exam. And this is probably where this is going to be hard. So given a table of amino acid differences, how can we actually make your own? And this is where this is making your own tree is actually hard. And I didn't want to actually explain it by myself. So I found this really cool video which had a bunch of animations that I'm going to play to show how to actually craft a tree. Alright, so let me know if you guys can't hear the audio.
What do we...
But I'll play this video for around 3 minutes.
Also is going to tell you about your out group. Now, species A and C, we know had 2 differences. Species C and B, we know we had 6 differences. So now we're at C to C, and it's like they're the same species. Then we have 5 and 9, and then we continue filling in this table. Now, what do we do with this table though, right? Like, how do we use this to draw our trees? So let's go ahead and check that out. So here I have the table, and what I like to do is I like to start with the one.
Okay, I'm going to pause for a sec. So this is the table. So by the number of amino acids differences, they're talking about the number of proteins are made up of amino acids, and 2 different proteins within 2 different organisms can give a signature way of showing by comparing these 2 proteins about how different these 2 organisms are by just analyzing their proteins. And so this is what that table is showing. It's showing that between species A and B, there's 4 amino acids differences. Between species B and C, there's 6 amino acids differences. And between D and B, there's only 1. And that's what this table is showing.
Ones that are most closely related. So what I see here, I also look for my outgroup. Which one is the most different is also very important. So here I have E as my outgroup. And then I notice that B and D have only 1 difference. So they have a very recent common ancestor. Then A and C also have a very recent common ancestor. And if you're looking at this, you might be thinking, well, that's not a cladogram or a phylogenetic tree. Well, for me, I find it really easy then just to connect the lines, basically. So now I have a tree, and if you check the numbers and data, it really does work out. Now, we also want to point out that these trees can be switched at the nodes. It would need the same thing. Now, we can also draw our tree this way. Oh, I'm sorry. So we can switch it at the nodes. All right, means the same thing. Now, if I look at the goalpost style, here B and D had only 1 amino acid difference, so they have a pretty recent common ancestor. And then we had A and C. They had 2 differences, so I drew their line, their more recent common ancestor, a little bit further back in time. If you remember from my last video, phylogenetic trees are kind of calibrated based on molecular clocks, like the rate of mutations and how many differences there are. Now, E was our outgroup, so that's a further back. So this is how I would draw this table in a phylogenetic tree style. All right, so that's it. I think at this point, if you're in a biology...
Alright, so that's pretty much... In order to review what she just said, basically, to make a phylogenetic tree, you're going to want to follow these not so basic steps, but barely basic steps. So first of all, you're going to want to find the outgroup basically if they ask you to make a tree, the first step you're going to do is find the outgroup, and that's the organism with the most differences from all the others. And in this case, as you can see in the table, that was species E because it's the most difference from the other organisms. And as you can see, as 10, 11, 9, 12, it's the outgroup. And then next up, you want to identify the organisms with the smallest number of differences. And as you can see, that's the differences between species B and D. And I won't go over this, but the reason why we do this is because we want to order the organisms in order in which they branched off from each other. A smaller difference signals that they branched off sooner in time than an organism that branched off with the larger amino acid differences, because they had more time to evolve away from each other. And then now you want to connect those two organisms first on the tree, and then you just repeat that process until you found until you finish the entire tree. And basically, is there a specific number for the smallest difference? I've also done around only three FRQs for this, but in the 3 FRQs I did do, it's always pretty obvious, and it's always either 2 or 1. But as a surefire way, it should always be, it's just the smallest one. But it will be pretty obvious that it's the smallest one. Like, you're never gonna have to decide between like 500 and 512. It won't be that hard. It will always be something like this. But for smallest difference, it's just the smallest. Does that make sense? Yeah. All right. So you're gonna continue connecting organisms in order of increasing genetic differences, and the more differences, the farther back in evolutionary history it was. All right. So this is I actually want to walk us through this together, because I actually realized that I was just doing this earlier, and it is a little difficult, so I'll walk us through this. But this is a modified 2018 AP Biology modified FRQ. Basically, the question asked, construct a cladogram from the table. And I'll walk us through this. So, as you can see, the first thing we want to do is find the outgroup. And in this case, there's only one obvious outgroup in this answer. And I think that we can all see that that's the panda. And then you want to identify the 2 organisms with the smallest number of differences. And that's the brown and black bear. As you can see, they only have 1 amino acid difference there. And so now you want to find, so that means that, first of all, at the base of the tree, as the outgroup, that's the panda. And the panda is at the bottom of our tree. Then, now we know our most recent one is going to be the black and brown bear, because they have the least number of differences. And then just by trial, well, you can continue on, but you can see that the polar and black bear would be next. And then, so once you know that, you can construct a tree like this. Yes, yeah, and she got it exactly. But yeah, as you can see, the panda is the outgroup. This is something I drafted up really quickly. The panda would be the outgroup. The black and brown bear, because they were the smallest differences, branch off the latest. And because, well, you can just do process of elimination. At this point, you can know the polar bear goes there. And this, again, usually AP tests will only have you do 4 different animals. So you can almost, like, they're never going to ask you to make a phylogenetic tree with 10. The most I've seen is 5. Yeah, I totally understand. Actually, there's another slide for this. They'll only ask you to do 4 animals. And now to explain this animal again, it's the amino acid differences in the list protein. And you can actually ignore the list part among bear species. This is just basically all bears have the exact same protein, and that's kind of implied in the problem. And it lists the amino acid differences between the, actually wait, how many of you know what amino acids are? Just to make sure I'm not, is anyone confused about protein structure? Okay, alright. Okay, so the amino acid structure, so proteins are all made of amino acids, and amino acids define what a protein is. So basically, it's saying that between I'll use the example of the black and panda bear. It's saying that there's a 33 amino acid difference sequence between the black and panda bear. And the same thing could be said for the brown and black bear. There's 1 amino acid difference between the black bear's protein and the brown bear's protein. And we can also use the example of the polar bear and the brown bear. There's an 8 amino acid difference between the brown bear's protein and the polar bear's protein. And I see where some of you are getting confused. It's because y is only half of the table used. And the reason that you only use half the table is because it would be kind of redundant to show more than half, because the difference between the polar bear and panda is already listed here. So they weren't listed up in the top right because it's already listed there. So that's why it's redundant. And the reason they have dashes is because it's just 0. I mean, the difference between the panda bear and the panda bear is 0 because they're the exact same organism. And the difference between the black bear and the black bear is 0. And the difference between the brown bear and the brown bear is also 0. And the difference between the that's actually something I also do, Fate. Yeah, I actually when I did this FRQ, I filled it in myself. It made more sense that way. But yeah, and then basically you can ignore the list part because this is what they'll always do on exam, whether it's DNA sequence or protein or whatever. They'll always give you a table detailing some sort of amino acid sequence or DNA sequence and listing the differences between them. And so back on how I'm gonna go over how to solve this again. So first of all, the reason why this method works is the reason why this method works is because you're finding the outgroup first, and then you're just consistently finding the one with the smallest differences, and then the second smallest differences, and just keep going down from there. And as you can see, black and brown bear, there's only one difference between them, so that's why they're at the top. Then between polar and black bear, that's 7, and then so that's why they're next. Yeah. And then panda bear, that's the outgroup, and that's why it's on the base. I know that this is super confusing. I know that this is super hard, so any questions about this? Yeah. Actually, I'll post this entire thing very quickly in it. Oh shoot, guys, I'm exposing my low level here. Yeah, guys. I know. Actually, wait, we'll actually report that real quick. Oh yeah, for sure. So I'll wait a couple more. Does that make sense? Or I'll also post these slideshows once this finishes in around I think 20 more minutes, roughly. We're almost there. Topic 7.9. Alright, so 1 can least diff, but then use brown as the longest line 1 because compared to black has least diff with the others. Oh no, it's okay, all good. I think I know what you mean. So you're saying that that between the brown and the black bear, it had only a 1 difference between them, and that's why they're at the top? And if so, yes, yeah, that's true. That's why they're at the top. Yeah, but then brown is most top because so I see what you mean. So you can actually rotate these nodes. So you can actually rotate the black and brown. You can swap the black and brown and you'll have the exact same answer. And the reason that is that nodes can be rotated, so the black and brown can be swapped individually, and the polar bear can be swapped with the entire pair of black and brown. So you can swap nodes. You can swap any node, and again, a node is where things branches. And so if you put down panda, polar, brown, black, that's also right. And if you put down panda, black, brown, and then polar, keeping in mind that you can rotate these nodes, then technically that'll also be right. No, yeah, no problem. So for the graph to read it, do you look at the two organisms and lower the number, the lower difference? Yes, yeah. By graph, I think you mean table, but the lower the number, the lower the differences in amino acids. Yes, exactly. Yeah, of course. All right. Well, if there's no more questions, I'll move on here. So that was pretty hard. I'll actually this is one of those things that requires a lot of practice, and you're essentially just applying this algorithm to some sort of table. And the reason why this algorithm works is kind of not intuitive, so it just requires a lot of practice. And I'll actually post the 2018 FRQ later in the slides, so you can take a look at it later. All right, I'll wait for any more questions. Yes, yeah, of course. I'm looking at the recording, the slides, and I'm looking at the CED too, and I'm looking some of my own study guides as well, and some of other progress checks. Yeah, of course. So, topic 7.10, speciation. Unlike phylogeny, there's a lot of stuff, but it's actually there's not that. Once I get into it, it won't actually be that hard. So again, I'll let you cruise it, and I'll post the CED in the chat. All right. So to start off, what leads to the formation of new species? And that's rhetorical. You don't answer that. So speciation begins when populations become reproductively isolated. And so what is speciation, right? It's when a single species rapidly evolves into two different species as a result of this reproductive isolation. And what is reproductive isolation too? So reproductive isolation is the process by which two or more populations diverge and become unable to interbreed successfully, leading to the new formation of a species. And this brings us to the biological species concept. And there's a few common misconceptions with this. A species is a group that can interbreed and produce fertile offspring. So what exactly does this definition mean? So a common misconception is that it's not really like what exactly it means. So a fish and a cat, a fish and a cat can't breed. A fish and a cat can't breed. All right. Without a better way of wording it. So because a fish and a cat are different species, yes, I will be posting the PowerPoint later. Yeah. And because the fish and cat can't breed together and produce fertile offspring, then that's why they're two different species. And that's how species are defined. Like that's the definition of a species, is whether or not two organisms can breed together and produce fertile offspring. And that's the definition of a species in AP biology. And that was a little confusing when I learned it a couple years ago. So if you have any questions, just ask me. So a biological species is a species that is a group that can interbreed and produce fertile offspring together. For example, a cat can breed with a cat and produce fertile cat offspring, but a cat cannot breed with a fish and produce fertile cat-fish offspring. Does that make sense? All right, perfect. And another thing, actually, I'll talk about that last bullet point later. All right, so how does reproductive isolation work? And so there's two main types of reproductive isolation, and that's prezygotic barriers and postzygotic barriers. So again, prezygotic barriers prevent fertilization. All right, so they prevent fertilization. Why does behavioral count when they can reproduce together? Okay, so behavioral, it's more of the fact that, actually, I'll finish first, and then I see what you mean. So it prevents fertilization, and postzygotic barriers, the fertilization occurs, but the offspring is inviable or sterile. An example of that would be mules when a horse and a donkey breed together. But in terms of your question, why does behavioral reproductive isolation count when they can reproduce together? And that should, in theory, mean that they should be the same species. And I think that's because hang on, let me think for a sec. It's because in the sense that they have different mating rituals. And so let's say that we have a bee, two insects that have different mating rituals. They will never mate each other in nature. And I think that's also a key part of that definition that's not listed. But a key part of it is also that they should be able to reproduce in nature as well. And that there's no human intervention with them. So why does behavioral count when they can reproduce together? And that's because it doesn't really happen naturally. And does that make sense? And I think that's a key that I should have put down in the CED. But yeah, it should be natural. And again, post-zygotic barriers, they occurs, but the offspring is inviable or sterile. So if we did breed a cat and a fish, sorry, I keep coming back to this example because it's the only thing I can think of, even though it's a little morbid. But we can't. If we did breed a cat and a fish together, likely the offspring would not survive. If that makes, yeah, it wouldn't survive. And if it did survive, it wouldn't be able to reproduce. Yeah, I agree too. All right, all right. So another thing, there's two different types of speciation. And the two types are allopatric and sympatric. And again, this is just something you got to memorize. But allopatric speciation occurs due to some sort of physical separation, but sympatric speciation occurs because there is no physical barrier. And so allopatric speciation is really straightforward, right? That's just because of physical separation. But how can sympatric speciation actually happen? Like, how can organisms become physically reproductively isolated, even if they're in the same area? And that's often because there's a behavior change. They live in the same area, but due to a random mutation or something, certain fish get preferred over others. In this example, like people would prefer to mate with behaviors change mostly through mutations or just some sort of random event that happens in a vacuum. Yeah, or people, yes. But yeah, and or okay, there's also, I'm thinking of another example, or the use of some sort of different resource that gets like introduced. Like if humans introduced a new food source into there, then their behaviors will change, and that can randomly lead to their reproductive behaviors being changed. And so that's how sympatric speciation will occur. But the most common type is physical separation, and that's allopatric. All right, so now there's the speed of speciation, and there's just two models for this, and that's all you need to know. That's gradualism, which is that changes occur slowly over time, as seen in the left graph. And there's also punctuated equilibrium, which there's rapid bursts of change after long stability. And another thing that I also want to talk about is adaptive radiation. So, is a special case where one species, of course. One rapid species rapidly splits into multiple different species to fill new niches. And that's an example of gradualism. As you can see, speciation. Okay, I'll give an example of speciation. That's when I'm gonna go back to fish. I'm gonna go back to cat and fish because that's the one thing on my mind right now. That's when a fish slowly gains different traits slowly over time, like a day, like one generation after the other, they always consistently gain a different trait. Punctuated equilibrium would be they go for hundreds of generations without a single change, but then randomly 2 generations later, they're completely different. And so that's punctuated equilibrium, and that's the difference between it and gradualism. Does that hopefully does that make sense, Greens? Or yeah, it's not really noticeable unless you have long periods of time. No, sorry, linear graph, that would be by adaptive, I think you mean adaptive radiation. So the rapid burst in this case would be the right graph. Okay, so the more accurate one is punctuated. Punctuated equilibrium, but And back to your question, is a linear graph adaptive? I think, well, no, punctuated equilibrium would be more adaptive because it occurs in rapid bursts. Let me know if that makes sense. Yeah, the right graph, which is punctuated equilibrium, is more accurate. Punctuated, but I see what you mean. Yeah. So I think they're talking about how it's linear in between. So as you can see in the right graph, it's flat and then it's linear, and then it's flat, then it's linear, but just fast. Is that what you're talking about? Um, okay. Uh, all right. Well, I'll move on, but I'll come back to you if you get that image. Oh, I see. Okay, I see. Um, dang, what the? Okay, I see. So, polygradualism. Okay, I've actually never heard of phylic gradualism, but in terms of punctuated equilibrium, I doubt you're going to see. I've never seen a graph like this before. In my in my 3 years of AV biology, but um, yeah, I don't think you need to worry about this too much. But I don't know too much about that graph you just sent. Sorry about that. Yeah, yeah. No, sorry about that. No problem. All right. So for the next, let's see. All right, yeah, that's the last slide before testing your knowledge. So all right, so a population of insects become, again, I won't use the timer anymore. I feel like 20 seconds is way too short. But a population of insects becomes separated by a newly formed river. Over many generations, the two groups accumulate genetic differences, and when brought back together, they no longer recognize each other's mating signals and do not interbreed. Which of the following best explains this outcome? Uh, all right, so I think that's everything, and I will go over why. So the two main answers here that could be confusing, A and B were all throwaways, but C and D were the distractors. So the reason, so the answer is actually C, but here's why D is wrong. So again, the answer is C because allopatric speciation occurs when a population is geographically separated, and in the case that was the river. But the fact that they no longer, and here's the key point, which could distract someone for why the answer is D, and that's why this is kind of hard because it's either C or D, is because the fact that they no longer recognize each other, mating signal, and do not interbreed, that is a sign, if alone, uh, signals that that's a reproductive barrier. And that's why the question said that after they were brought together, they do not interbreed to show that they were reproductively isolated. But I do see how people could see it's D. I mean, I'd also think it could be seen as D because if they're just together and their behavior changes, then that would be an example of sympatric. But in this case, C would be the better answer because they were separated and then brought back together, and they no longer reproduced with each other. Does that make sense? If any questions? All right. All right, guys, we're almost there. Our extinction. This one is actually probably one of the easier subunits. But all right, so again, I'll post CD in the chat, and I'll let you read it for a sec while I do that. All right, so topic 7.11 focuses mainly on extinction and what happens when an entire species or an entire population disappears. And it's just a natural part of evolution that can also open the new doors for evolutionary paths. I mean, uh, yeah, extinction isn't always bad. I mean, unless it happens to us, right? But, all right, so extinction is the complete loss of all individuals in a population or species. And this can be caused by environmental changes, the habitat loss, or some sort of competition. And as this occurs, I'm bringing back adaptive radiation because adaptive radiation is a rapid evolutionary process where a single species rapidly evolves into a new species. And the most common scenario in which you'll see adaptive radiation on a test is when they ask you what happens after a mass extinction. And the most common cause of a mass extinction is adaptive radiation. Example is the dinosaurs. As soon as they went extinct, life completely changed because new niches, ecological niches, and just to recap, niches are essentially different resource sources within an ecosystem. And so after a mass extinction event, niches open up, which allows for adaptive radiation to occur and evolve into these into multiple different species. And that's why extinction or speciation are rapidly linked. It's like life and death in a sense. Well, it is life and death. Extinction and speciation go hand in hand. All right, so I'm gonna move on. Any questions about this? All right. So that was pretty. There's only 2 more left. It ends at 7.13. I'll let you guys read this. This is 7.12. Variations in population. So it's all about why genetic diversity matters. And I think someone asked a question in the first 10 minutes about why genetic diversity matters. And the reason is populations with more genetic variation are more likely to survive environmental change. So a high genetic diversity means a population has more potential responses to environmental stress. No. I just put them there as because as a student, I remember I really liked self-studying some stuff. Oh yeah, I see. But in terms of learning objectives in the CED, I wouldn't. Yeah, I know. PST is kind of cooked. But in terms of learning objectives, yeah, I wouldn't focus too much on them because I'm covering essentially what you need to know in the CED. But it's the reason I put them there is just as I always want to give a general overview of what I'm covering before I actually go in depth in it. But yeah, I wouldn't focus too much on the CED. Just give it as an overview. Yeah. But okay, so back to this side. Oh yeah. Yes, yeah. So, the LOS. Wait, sorry, what does that stand for? But yeah, when I'm making these slides, because I took AP Biology quite a bit ago, and so sometimes they change their CED. So, oh yeah, learning objectives. Yeah, yeah. All these slideshows are based off of the CED because sometimes they change the CED year by year. I don't want to accidentally include something that I learned but is no longer in the CED. So that's why I always reference the CED. But yeah, back to this. Oh, sorry. Yeah. Does that make sense? Yeah. All right. Great. All right. So, populations. If a new species or disease or climate event hits, it's more likely that at least some individuals will have the traits that help them survive. So, in contrast, low diversity populations are more vulnerable. There's a less chance that someone has the right traits to handle the change. And to give an example of this, given back to the 3-person example, let's make it 2 people. Let's say that one person can breathe underwater, and let's say 100% of the population can breathe underwater. Given an event where a huge drought, let's say the oceans get drained, all those people are going to die. But in a genetically diverse population where 10 people can breathe above water and 90 people can breathe underwater, in the event that a drought occurs, 10 of those people will survive. And that's what we're talking about by high genetic diversity means a population has more potential responses to an environmental stress. Basically, if a new catastrophic event hits, they'll be more likely to survive. All right, so adaptive versus deleterious event alleles. So an allele can be beneficial in one environment but not harmful in another. An example is a rodent with light fur blends better in sand environments by hiding them in darker environments. Sorry, no, it doesn't help them. A rodent with light fur blends better in sandy environments than blending in dark environments. So that's why an allele is good in one environment but harmful in others. And that's why selection pressures vary with the environmental context. All right, so any questions about the slide so far? All right, if not, I'll move on to the next test your knowledge. All right, so again, I won't use the timer, but which of the following best explains why genetically diverse populations are more likely to survive environmental changes? Oh, congratulations. All right, I think, all right. So yeah, I think we all got that right. So this is just a direct. Basically, more genetically diverse population means more individuals that can survive it. Because they possess a wide variety of alleles, which increases the chance that individuals possess certain traits that help them survive this changing condition. Yeah. All right, next up. Oh, the video plays. All right, so shoot, sorry. All right, so guys, well, it's been an hour and a half, and this is the last one, and thankfully it's incredibly light. So I'll post this video in the chat. And again, the reason I post these here is so just to give a general overview of what I'm covering before I go too deep in it, and it also helps know exactly what you need to know for the exam. Make sure you don't learn anything extra. But all right, time to go too deep. Time to go in depth. All right, so the timeline of Earth. So let's be real. The dates are just so solid. This is all in the CED, but I've personally never seen a question that asks you to say, "Oh, when was Earth formed?" So this is on the CED. This is on the CED, but I've never seen a question that asks for it. So you just keep this in there, but it's not really major. But you also need to know that organic molecules could have been transported to Earth by a meteorite or some other celestial event. And so if you see that in MCQ, just don't immediately throw it away because it could be true. Yes, these values are on the CED. It does say Earth formed approximately 4.6 billion years ago. The environment was too hostile for life until 3.9 billion years ago. And the earliest fossil evidence for life dates up to 3.5 billion years ago. BYA stands for billion years ago. So these values are on it, but I've taken so many tests, I just don't really see it. I haven't seen it, honestly. All right, but here's what you do need to know. That's how life got formed. So there are three main conditions that made life possible on early Earth, and that's inorganic molecules, or precursors, were available for the synthesis of first organic molecules. The presence of free energy drove chemical reactions, and that's just free energy. By free energy, I'm talking the sun, just like random energy that's around, pretty vague. But here's the trick question. I remember, I still remember this. There's an MCQ on this. There was an emphasis on absence of atmospheric oxygen. Oxygen back then was actually harmful to life because the absence prevented oxidation of organic compounds. And so here's how I like to remember these three conditions because you will need to know these three conditions. Icy Earth's atmosphere, inorganic precursors, energy, and the absence of atmospheric oxygen all contribute to the formation of life. And again, icy because I is inorganic, Earth's energy, absence of atmospheric oxygen, atmosphere. So yeah, you will need to know these three factors, and I have seen it tested. All right, and next up is the RNA world hypothesis. The RNA world hypothesis essentially states that RNA came before DNA as the original genetic information, and this is because RNA can store information and it can replicate itself, which makes it a strong candidate for the earliest replicating molecules. And this idea just explains how life could begin with just a single type of molecule before more complex systems evolved. All right. So any questions about this or any of the previous slides? All right, sounds good. And so that concludes the full Unit 7 review. So yes, natural selection. Oh yeah. I got questions. Yeah, what's up?